Hello students, myself Popere KS, Assistant Professor at Ray Shikshan Sasthas, RBNB College, Sri Rampur. This is a practical for FY BSc. So this is the first practical based on, that is BO 113. It is based on the theory syllabus, that is BO 112 and BO 113. In this practical, the aim of this practical is to study the life cycle of the Spirogyra. So in this practical, it will be consisting of the that is the thallus structure of the Spirogyra and about the reproduction of the Spirogyra. So firstly we will see the systematic position of Spirogyra. Division is Chlorophyta, class Chlorophyceae, order Conjugales, family Zygnumetaceae, and genus is Spirogyra. Now this Spirogyra, it is a free living algae. It is mostly live in the fresh water like ditches, river, and even in the that is all types of the fresh water. It is an filamentous algae which is living or freely floating on the surface of the fresh water. So this is also called as pond scum, and there are different species of the Spirogyra which are found in India. About 23 species of Spirogyra are found in India, that is Spirogyra cylindricum, Spirogyra variabilis. So different species are there. And so now this is the procedure of the practical. In this, first we are going to take the material, that is the Spirogyra. Now it is present here in the bottles. So this is Spirogyra. This is the material for the Spirogyra. Now if we take the material in the Petri plate, the material will look like this. So this is the material of the Spirogyra. It is a filamentous structure. So this will be seen in the form of a filament. Now what we have to do is we have to take the material on the slide and put it under the microscope. So what we are going to do, firstly we have to take a very little amount or a very small amount of the material, that is it should be only one filament on a slide. So the filament first should be separated. Now take this on the slide. Here also on the slide, separate the material as much as you can. Now remove the excess. Now this slide, it is having a very little amount of the material or a very small filament. Now take a drop of safranin . Now we have taken a safranin. Now take a cover slip and put the cover slip. So this is how, this is a slide and on which I have put in the cover slip. Now take this slide and put it under the microscope. Set it under microscope. So this is how the slide should be prepared, and at the microscope it should be focused here. So now we can see the filaments of the Spirogyra. For this, what you need is, you should have a well equipped that is the dissection box. Okay. Now after the procedure, that is, we have prepared a slide under the mic and seen it under the microscope. Now we are going to see what is we have seen under the microscope, or what is the material which we have put it under the microscope. It will show this, that is a filamentous structure of the Spirogyra. Now I am going to explain you the filament of the Spirogyra. Now this Spirogyra, this is a single filament of the Spirogyra which is seen under a microscope. So firstly, it is filamentous and green in color because of the presence of the chlorophyll. Now next to that, it has a presence of cell wall, that is the outer cell wall, and it is made up of pectose. So because of that, this Spirogyra, it is slimy to touch because of the presence of the mucilaginous sheet that is made up of a pectin. Now next to that, the filamentous, they are multicellular, made up of a different cell. Now each cell, it is similar in size. In the filament, the two cells, now these will be similar in size. Next to that, now in each cell there is presence of a spirally arranged chloroplast. Now these are the spirally arranged chloroplast. It will be present throughout the filament in each cell. It also has presence of, now on this chloroplast there are pyrenoids are present. So these are the pyrenoids which are present on the chloroplast. Next to that, it also has presence of nucleus. So each cell will have a nucleus that will be present in the center, and it is held here by the cytoplasmic strand. So cytoplasmic strand in each cell, it will hold the nucleus. So this is about the filament of the Spirogyra. Now we will see the reproduction in case of the Spirogyra. Now this Spirogyra, in the reproduction, it takes place by two methods, that is asexual method and sexual method. So firstly, the asexual method, it will be takes place by the means of fragmentation. Fragmentation means what? This fragment of the Spirogyra, it will break down into two pieces. Now the two separate fragments, it will be acting as a new plant body. So that is the asexual reproduction. Now next is sexual reproduction. This sexual reproduction, it also takes place by two methods, that is scleriform conjugation and lateral conjugation. So sexual reproduction, it is by means of conjugation, and there are two types of conjugation. So let us first see the first conjugation, that is the scleriform conjugation. Now in case of the scleriform of conjugation, there will be involvement of two different filament. So like you can see here, there are two filaments, they are involved in the reproduction. Now these two filaments, they have different polarity, or they are of different stain. Different stain means one here, it will be of a positive stain, and the other filament, it will be of a negative strand. Now what will happen? These two strands or the filaments, they will come side by side together they may come throughout the filament or only a part of the filament that can become side by side. Now what will happen? Now from the two filament, that is from the both of the filament, there will be appearance of a structure, like you can see here. The first, there will be a protuberances like structure will be forming from both the filament, and after that, so this is called as a conjugation tube. Now this conjugation tube, it will increase in size, and both the tubes, conjugation tubes of the two filament, they will be joining, or there will be a formation of a conjugation canal. So one canal will be formed between the two cells of a two different filament. Now what will happen after this process? There will be, now this positive suppose is acting as a male filament and negative is acting as a female filament. So what will happen now? From this side, that is from the positive strain filament, the cytoplasmic material as well as the nucleus will flow towards or move towards the negative strand or the negative filament, and after that what will happen? All the material has gone from this cell to this cell. Now what is here? This cell, that is the positive cell, it is empty, and this will later on maturity will form a zygospore. So this is a zygospore, that is the zygote, it will be formed. So it will be in the case of a scleriform conjugation. Now next is lateral conjugation. In case of the lateral conjugation, as in scleriform, two filaments were required, but in lateral conjugation only one filament will be there, and reproduction will be takes within the same filament. So now here you can see, here will will also protuberance like structure will appear at this side, that is from the two cells, both the cells will forming a protuberances towards the end, that is toward this strand. Now later on the wall between these two cells, it will dissolve, and the material, now here also there will be positive and negative strand. So the two opposing or the two side by side cells, they will be forming, one is acting as a positive and next is acting as a negative state. So from positive to negative state, there will be all the material, that is a cytoplasmic and the nuclear material, it will be flow from one cell to other cell. Now after that, the one cell will become empty, and here there is a formation of a zygote or the zygospore. So till here this two types of the conjugation, that is the from till this stage there is a difference. Now after this stage, that is a zygospore stage, what will happen? Now the each filament, it is an haploid, that is NN. After the this conjugation, it will become 2N, that is diploid. So zygote is the only stage in case of the Spirogyra which is diploid. Now after that, what will happen? There will be fusion of this nucleus. Then after that, there will be reductional division due to which there is formation of a, so due to that there will be formation of 4, that is a 4 nucleus will be formed in this cell. Now after that, what will happen? Of this 3 will degenerate and only one functional will be there. So these 3 are lost and only one functional nucleus will be there. Now after this stage, what will happen? This will grow into a new haploid filament. So after that, the zygospore, it will be developed into a new haploid filament. So the life cycle of the Spirogyra is haplontic, because the filaments are haploid, and after this stage this cell is also haploid, and haploid is again forming the new filament. So this is about the life cycle of the Spirogyra. The only difference between the two conjugation is that it will involve the involvement of 2 filament is there, and in lateral conjugation only 1 filament is there. So this is about the observation which will be we observe under the microscope. So after that result, that whatever we are going to see under the microscope, we will put it under the result that the filament were seen, or the type of which the one of them or any two types of the conjugation or the reproduction which will be seen under the microscope. Thank you.